And give us one moment. It is setting it up to go live, and then I will make a couple of quick announcements and introduce Bill Parker. Good afternoon. Thank you so much for joining us for the July 2024 edition of Lunch a la carte, a monthly Zoom meeting, a lunch and learn, if you will, where we get an opportunity to hear from one of our previous grant researchers and to hear a little bit about the state of research in the field of Alzheimer's and dementia. We are so excited that you are joining us today, either live or on YouTube on a replay. I'm Tiffany Irvin, the Executive Director of CART, and we are honored to be able to present this program for you guys every month. I have a couple of quick announcements, and then we're going to move directly into our presentation today. I want to remind you that the CART Fund hosts 2 meetings every month via Zoom. And one is this lunch a la carte, which is the date does change a little bit every month due to whatever our researchers' schedules are. We want to make sure we're amenable to them. The other meeting that we host is on the third Thursday of every month from 5:30 to 6:30 p.m. And that means that will be tomorrow night. And we call that CART Conversations. And everyone is invited to join us for that meeting as well. We talk about fundraising and public image and the state of CART and our grants process. And we also have a lot of really helpful tips that will help you either in your Rotary Club, in your business or civic life, and of course, in what you do to help us promote the CART Fund. So we want to make sure that you join us for those meetings if you are able as well. We also have a new Rotary Club that is forming, and they are a cause-based Rotary Club that is specifically focused on Alzheimer's and dementia research, and they meet on the third Tuesday of every month. And you can also find them and their information on our website or in the DACTV calendar if you are a member of a Rotary Club. So lots of exciting events coming up. The next Lunch and Learn, the next Lunch a la carte will be on August the 21st with Dr. Levi Wood from Georgia Tech. And we want to make sure that you go ahead and get that date on your calendar as well. And now I'm going to turn this over to Bill Parker, who is a past president of the CART Fund, and will introduce today's speaker.
Thank you very much, Tiffany, and Dr. Leissring, welcome. We are delighted to have you. For your intro, Dr. Malcolm Leissring is an award-winning biomedical researcher who is internationally recognized for his work on molecular pathogenesis of Alzheimer's disease. He conducted his postdoctoral training at Harvard Medical School in the laboratory of Dennis L. Selko, medical doctor, one of the world's leading Alzheimer's researchers. Dr. Leissring has held faculty positions at Harvard Medical School, the Scripps Research Institute, Mayo Clinic, and is currently at UC Irvine's Institute for Memory Impairment and Neurological Disorders. His research has been funded by grants from the National Institutes of Health and various private foundations, such as the Alzheimer's Association, the Ellison Medical Foundation, the American Federation for Aging Research, and our own Coins for Alzheimer's Research Trust. In 2009, he received a CART grant of $200,000 while he was working with Mayo Clinic. Dr. Leissring, welcome. We are delighted to have you. We are delighted to hear how you've been doing since 2008 when we met you in Columbia, South Carolina.
Yes, it's been a while, hasn't it? It's amazing to think it's been, 15 years since the since originally getting this. And I thank you, everybody. Thank you for the welcome and for the introduction. And I'm really excited to be here. And I have since become a kindred spirit of sorts. I started a nonprofit called the August Dieter Foundation, named in honor of the first person diagnosed with Alzheimer's disease. That's focused on Alzheimer's prevention because, as we all witnessed, we went through a generation of clinical trials. If you were financially savvy and you had bet against every clinical trial, you'd probably be a trillionaire by now, because it was at about, 274 out of 275 failures at one point. Now, I know there's some things that have, shown some promise, but they have complications too. And I still think prevention is the best way to go. So I'll mention it at the very end. I've got a website if you're interested. And I was selling a dietary supplement that was shown to prevent Alzheimer's in animal models, consisting of nothing really extraordinary. So I do think the earlier you start, there are things out there that are natural and safe that can do as much as these expensive therapeutic drugs that have risks. So, all right, so I'm going to begin by sharing my screen. So let's see. So I'll go here. Okay. So I was looking for the perfect present, trying to think of how could I do this presentation, and it was remarkable. I actually found the old one, and I'm like, I did a good job back in, I think it was 2012 when I did this. And I just wanted to mention a little anecdote. It just occurred to me this morning. I remember my dad sent me this gold chain, and he goes, "This will be good luck for you." And I put it on, and that day I got a call from Bill, I think. And it's like, "Hey, we're going to give you," I think it was something odd, like you had a little extra, but not enough for a full normal grant. And so you went with 200,000. That was fantastic. So it was really, really a great thing. Okay. So I'm going to be talking about what I often call the forgotten side of Alzheimer's disease, which will make sense in just a second. So I know you guys have probably seen dozens to hundreds of these talks, so I'm just going to go through quickly to make sure everybody's on the same page. But the principal pathological features of Alzheimer's disease include these so-called plaques, which are extracellular and are what we call an invariant feature of Alzheimer's disease. You can't have Alzheimer's disease without plaques. But then there are these intraneuronal, intracellular, inside the neuron tangles, which are composed of phospho-tau. And, interestingly, these are common to other types of dementia. And, we've had lots of animal models that have plaques without tangles, and they don't seem to have the same symptoms as true Alzheimer's patients. So this is a very important part of the disease, and is probably, I like to say that excessive amyloid accumulation is a sufficient precursor, or it's like a trigger for the proximal cause of neurodegeneration, which is really these tangles. And then I'm going to add an extra thing here, which is lysosomal dysfunction. 50 years plus ago, it was widely recognized that something was going on with the lysosomes. They were swollen and all sorts of problems. And so lysosomes are these intracellular compartments. They're contiguous with the extracellular space, so that, you can go into them without passing a membrane. And these are intracellular acidic compartments, basically what I'm going to call the garbage can of the cell. And this is also an invariant feature of AD that just gets overlooked quite a bit. And then, of course, all this leads to neurodegeneration. And I'm realizing I just need to define the peptide in the plaques is amyloid beta protein. Okay. Or A beta. So quickly, this is what I'm going to call the life history of the amyloid beta protein, which starts off as a piece of a larger protein called the amyloid precursor protein, or APP. And then it gets removed from this by the action of two scissor-like enzymes that have been named secretases. And that's a really interesting point, as you'll see as we progress. they were called secretases because the beta amyloid peptide was known to be secreted into the extracellular space, and also, plaques are extracellular, so that's how they got their name. So beta secretase has been identified as this enzyme called BACE, BACE1. And then gamma secretase is a very unusual enzyme, which we call a protease, because it cuts right in the middle of the membrane, which I'm showing here. And when you cut a protein, it requires a water molecule, and yet that's inside a fatty bilipid layer. So it's very kind of unusual. And one of the features about this so-called gamma secretase protease is that it's sloppy and it cuts at multiple sites. So you get beta amyloid that's 37 amino acids long, or 38, or 40, or 42, or 43. And this is important because the longer peptides are more likely to aggregate, more amyloidogenic. And they're sort of, you could think of it as like there's good cholesterol and bad cholesterol. It's sort of like the bad amyloid is the longer amyloid. And, I get so tired of seeing these, it seems like one of the favorite tropes in the media is that the amyloid hypothesis is dead and we wasted all of our time studying this. It's just the most ridiculous thing ever if you realize that there are about 500 different mutations in the amyloid precursor protein, or these enzymes that cut A beta here, particularly gamma secretase, that all cause Alzheimer's disease with 100% certainty, and that happens early in life, very aggressive forms. So and all of these affect beta amyloid or amyloid beta metabolism. It somehow affects it. It's either giving it more, or it's shifting the ratio of the longer bad amyloid to the shorter good amyloid. So you just cannot deny that amyloid perturbations cause Alzheimer's disease, because they very much do. Okay. So once the A beta gets secreted, as they say, it aggregates and then we lead to toxicity. And this is basically where we were stuck, in my opinion, for many decades. Like, this is the end of the story. It gets produced and it forms a plaque and that's it. Not at all. As a matter of fact, the vast majority of beta amyloid is degraded. I think I did a back of the envelope calculation once, obviously very rough, but it would, over 8 decades of life, you would probably produce around 20 pounds of amyloid. So nobody has 20 pounds of amyloid in their head. It's normally being degraded and or removed. And this is important, this clearance, because I'm putting circles around it, because it goes inside cells. And guess where it goes? It goes to the lysosomes, in particular, where it can be degraded. So I think the field had been focused more on the devils, because, you can develop an inhibitor and, theoretically cure the disease, and ignoring the angels that were staring us in the face, which are sort of opposed to the secretases. So it leads to this view of there being a balance between the very few factors, only 3 things on the A beta production side, versus degradation, which actually has multiple players. So, the way we go about assessing the relative importance of these typically is we'll knock out, say, a gene for one of these proteases and see what it does. So we knock it out and then we look at, is there more amyloid or, unchanged? How do they compare to other ones? So here's an example of, it was the first direct measurement of the effect of these proteases on the removal of A beta. The A beta catabolism is the opposite of anabolism. It's the degradation. So these are mice that are lacking neprilysin here in the open triangles, which is a protease. And they have roughly twice as much A beta as in the extracellular space, the so-called interstitial fluid, as animals with 2 copies of neprilysin. And then we hit this with a gamma secretase inhibitor, which stops all production of amyloid. And you can see that it drops off right away. And the interesting thing is that when you calculate the half-life of the amyloid, it's a little bit longer in the absence of neprilysin. So it sort of goes to show you how important these proteases are. So what happens if we boost these things? So can't we do something like that, like remove the, lower the amyloid quite a bit? Well, in fact, in 2003, I published on a transgenic mouse overexpressing the neprilysin gene about 8-fold above normal. And you could see that this overexpression completely 100% prevented all amyloid plaque formation in these animals. This is more clean of a result, or more thorough of a result than any of the antibody treatments. I want to impress on everyone that there are multiple A-beta degrading proteases, and they're sort of listed here. Insulin degrading enzyme is one that I have focused on since my time at Harvard, and actually just published a chapter in the Handbook of Proteolytic Enzymes on that. And I developed the first inhibitors of this enzyme, and they have more utility for, you would think, diabetes. And we had a Nature paper in 2014 showing that they have anti-diabetic properties. I'm going on this tangent to explain why there was kind of a delay between some of my return to Alzheimer's. So I also developed inhibitors that are topical, and they actually stimulate collagen production. And so I recently started a publicly traded company and have a patent on them, and we're trying to go into the aesthetics and wound healing spaces with that. So, I mean, obviously these enzymes don't exist only to degrade amyloid, but they do have diverse features that I think is worth highlighting. So, for example, functionally, we have what I would call physiologic regulators, such as neprilysin. You can see, you knock it out and you get an effect. However, there are some proteases you knock out in a normal animal and you don't see any effect, but they only become into play when there's pathology present. So I call those maybe pathological regulators. Then there are perhaps therapeutic regulators. You could have a protease that doesn't do anything when you knock it out, but it might have utility as a therapeutic protease. And then I think, from my own perspective, I think these are very, very important experimental probes to address specific hypotheses or questions about how Alzheimer's actually happens. Okay, then you have things that are peptidases and proteinases, and they degrade oligoamyloid or fibrils of amyloid. And then you have ones that cut in the middle, and they cut on the end or on the other end. So there's really quite a wide variety, as opposed to the rather limited set of things on the A-beta production side. So let's just to highlight these asymmetries, as I call them, and I'll call this the hypocatabolism hypothesis of Alzheimer's disease. They did beautiful, elegant studies in around, when was it, 2008 or something, where they assessed the production and degradation of amyloid in awake human people, and people with Alzheimer's disease had a reduction not in amyloid production, but in amyloid catabolism, so in the removal of amyloid. So, unlike the 0.5% of people who get Alzheimer's due to those mutations that affect amyloid, maybe the other 95.5% are attributable to defects in the removal of amyloid. So there's many more players, as I've said. So this may account for a large fraction of AD cases, may help account for the so-called polygenic nature of AD. It's a complicated disease, and it vastly increases the number of drug targets. So they can be therapeutic targets and therapeutic agents. And interestingly, they're vulnerable to aging and environmental insults. So it might explain why aging is the #1 risk factor for Alzheimer's. Maybe these things break down or get oxidative damage, and that would explain why they're involved in Alzheimer's disease. Now, the important point I really want to highlight is this one, that different proteases will affect what I'm going to call different pools of A beta that might be differentially involved in AD pathogenesis. So if you look here, here's a couple of compartments of the cell, like the cytosol, the nucleus, you have extracellular space, then you have mitochondria, peroxisomes, endosomes, lysosomes, the endoplasmic reticulum. These are all different compartments. And if you look at where amyloid is produced and accumulates, it's kind of limited to these ones on the right here. And the field, in my opinion, has emphasized the extracellular ones because there's a certain dogma that it's secreted peptide that and the extracellular forms are what's most important. But if you look, there's a couple of these that have an unusual localization, including this one at the bottom here, which is cathepsin D, which I'm going to come back to in the second half of my talk. It's located in the lysosomes, unlike quite a few other proteases. Yeah, it's extracellular and it's cytosolic, but it's not really active in those compartments. So we sort of have now a clean way, if you study an enzyme that's only extracellular, like neprilysin, or one that's in this acidic compartment, it's more intracellular, you can maybe assess the different importance of those two pools of amyloid beta. So what CART helped us do is address this question, or are there any more of these things that remain to be identified? I showed you a long list. Absolutely, there are more. And to demonstrate that, I'm going to show on the, this is the % increase in amyloid in mouse models when we knock out different known beta amyloid degrading proteases. I didn't show cathepsin D here. But here's animals that were treated with one drug that inactivates just one category of proteases. And you can see there's a huge increase that is not matched by any of these. In fact, I'll show you some combinations of these, like neprilysin and IDE knockout. They don't get you anywhere near as close as or as high as a single drug will. Now, so what the CART grant funded was a search for novel A beta degrading proteases, A beta DPs here. And the results are, or the technology is not so important as it is to say that here are the results. And not every protease in the entire genome was there, but there was a solid, 600 or 700 that we analyzed. And there was a true winner in this, which is, you may have heard of BACE1, which is the protease that makes the initial cut to remove amyloid from amyloid precursor protein. Well, lo and behold, its cousin, BACE2, actually, when overexpressed, dramatically lowered the amount of A beta in the medium. And we confirmed when we compare these two, we see no effect of BACE1 on amyloid expression in this system. But BACE2 was very, very effective. And we went on to compare it, like we took cells and we transfected them with either BACE2, this newly identified protease, or some of the leading candidates that are out there. And you can see that BACE was, as effective as anything else. Okay, and so, that's mostly what this grant funded, and there are other leads that could be followed up on. And now I'm going to switch to some results that we've published recently that focus on a protease I mentioned before, cathepsin D. So what's really interesting is that cathepsin D is a relative of both base 1 and 2. There's like these three guys are all have a very similar, you know, they're related to one another. And when they were developing drugs that target beta secretase, base 1, the main challenge they faced was to not target cathepsin D, because the active sites are so similar that it was a real big challenge. So this protease lives in the lysosomes, and it's an aspartyl protease, just like beta secretase, just like gamma secretase. These work best in an acidic environment, like in the lysosomes. So there's a common variant in one amino acid in this that has been linked to Alzheimer's disease genetically. And in fact, a meta-analysis shows it's pretty comparable to all these other ones that people are so excited about. But for some reason, people didn't really focus on this. And more recently, mutations have been shown to be involved in multiple neurodegenerative diseases such as Parkinson's disease, which shares some common pathological features. So, but here's the thing. In 1996, cathepsin D was found to be the major A-beta degrading protease in brain extracts. And guess what? A year later, it was shown to degrade tau, which makes up the tangles that we all know and love. And nevertheless, very few studies had investigated cathepsin D mediated regulation of A-beta or tau in animal models. And I think one of the reasons for that, besides the general bias of the field towards production, is that for a couple years there, cathepsin D was a leading candidate for base 1. It was a leading, or for beta secretase. It was a leading candidate for gamma secretase. So people always thought about it as maybe involved in the production. So and then once we found the other actual mediators of beta and gamma secretase, you know, the damage had been done. Okay, so we did kind of recapitulate an experiment that was done in 1996 using cathepsin D knockout animals I got from Paul Softig. So they have no cathepsin D. And here's the key thing. We had to look at different pHs because, well, when you look at different pHs, so that we just took brains from wild type normal animals or cathepsin D knockout animals, and you grind them up and you test beta amyloid degrading activity in them at different pHs, you get two peaks in the wild type animals. A smaller peak at neutral pH, and then a huge one at acidic pH. And this one at neutral pH turns out to be insulin degrading enzyme, which you can show by adding insulin, which rather selectively blocks it. So you can see this lower thing basically shuts off almost all degradation at neutral pH. But this gigantic peak, shall we call it, is entirely attributable to cathepsin D. And this cuts in the same exact places where base 2 cuts, interestingly. These are the amino acids that make up beta amyloid. And I just want to emphasize, remember, we usually, when there's a bad amyloid, which we usually focus on A beta 42, which has these 2 extra amino acids. And then I would call it the good or neutral amyloid, which is A beta 40, which lacks those. So this is going to become very important, as you'll see. So when we looked at levels of 42 and 40 in these animals, now one thing about these animals, cathepsin D knockout mice die prematurely. They die at around 27 days of age. You can distinguish them from their litter mates only at around day 23 or so. So they kind of fall off really fast. They die from a peripheral kind of anorexia from their damage to their intestines. So we decided to look at a range of ages of animals just to be sure we weren't fooling ourselves with some kind of, accidental effect on amyloid. And what we saw was very large increases in A beta 42 and also large increases in A beta 40 that kind of rise over time as the animals age. So then we compared these to mice that were lacking neprilysin, which is considered one of the major A beta degrading proteases, or IDE, which is also considered an A beta degrading protease, major A beta degrading proteases, or mice lacking both of them. And you can see that cathepsin D just completely outshine both of these in terms of their effect on amyloid. And I want to emphasize this is insoluble A beta, which is really the stuff that is the bad stuff. This is the aggregated kind of form. And IDE actually has no effect on that because it can only degrade soluble forms of amyloid. Okay, so I mean, look at this. This is something that had never been seen before. These are 3-week-old animals that have mouse beta amyloid. Mouse beta amyloid doesn't even aggregate. It doesn't have any tendency to aggregate in the way that human A beta does. And we're not overexpressing it like we have all these transgenic mice that overexpress amyloid to like 100-fold. And yet we see frank deposition of amyloid by 3 weeks of age in these cathepsin D knockout animals. So, of course, I thought this was a stunning result, but the reviewers thought that I should test it in amyloid precursor protein overexpressing mice. It's just a more severe form of the same thing. And you can see the intense aggregation in this, what I'll call a perinuclear, around the nucleus manner. And that's basically where lysosomes live. And so that makes sense. And there were no effects whatsoever on extracellular amyloid. So we have the largest increase in total brain A beta we've ever seen when we knock this protease out. And all of it is intracellular. And those intracellular compartments make up just a tiny, tiny volume of the brain. So this is really actually a dramatic result. Okay. So now, if you notice, the increase in 42 is like higher in percentage terms than the 40. And when we look at this thing called the A beta 42 to 40 ratio, because mutations in gamma secretase, that's what they do. They increase A beta 42-40 ratio. They change where that enzyme cuts in the middle of the membrane and cuts the second cut of amyloid to produce amyloid. And these animals had a very consistent, highly statistically significant increase in A beta 42 versus 40. And we don't see that at all in these neprilysin IDE knockout or double knockouts. Well, that was interesting, and I thought this would be a very complicated mechanism. But when you discover a protease, the first thing you do is kind of characterize it, right? You want to know, the enzymatic properties, as we call it. And this can be a very arcane thing but I'll try to express it. So basically, the way you characterize a protease is you add increasing amounts of the substrate, amyloid beta 42 in this case, okay? And then you monitor the rate at which it cleaves those, right? So when it's really, really dilute, it starts down here at 0, right? Because there's almost no substrate to find. And then you increase it until it hits kind of a plateau. And that's really the maximum limit. You could deduce the maximum limit of how efficiently the protease can degrade this. So for A beta 40, this looked like a very normal garden variety A beta degrading protease. It it has an affinity. We measure the affinity, like how strongly do the two things come together. That's in this range. And it's a fairly efficient enzyme. You know, one molecule processes 21 molecules of A beta 40 per minute. But if you look at A beta 42, first of all, this affinity is around, 100 times stronger. So A beta 42 binds way more tightly. They find each other way more rapidly. And look at how slow that so-called turnover number is. It takes one molecule of cathepsin D about 4 to 5 minutes to process one molecule of A beta 42. So this is really remarkable. And I was really lucky to be at Mayo Clinic when I did these experiments because we had freshly generated monomeric, non-aggregated A beta 42 and A beta 40. It's the only way you could really do these highly quantitative measurements. And so these are really, really, truly accurate results. But we did verify it by other means. This is kind of an arcane thing called surface plasmon resonance, but it really is an effect. That it's genuinely a thing that's happening. And the way I sort of emphasize this is you see this blue line here and the scale of this graph. Well, this dotted line represents where this whole graph here would fit on this same scale. So they're clearly very, very different, despite we've never seen this before with another protease. We've not seen this diverse difference between 42 and 40. So when you take those two things together, like a stronger affinity and a slower processing time, what that ends up doing is making this A beta 42 a inhibitor of cathepsin D. So it degrades, it blocks cathepsin D. It's so busy processing A beta 42 that it can't do anything else. And so we quantified this by, putting different amounts, different concentrations of A beta 42, and then monitoring cathepsin D activity with a different kind of assay. And if you look here, 3 nanomolar of A beta 42 is all it takes to inhibit 1 nanomolar, so like 3-to-1 ratio here, by more than 50%. So, I mean, this is a ridiculously strong interaction. Whereas if you look at A beta 40, which is just missing 2 amino acids, this is 3,000 times less effective at competitively inhibiting. So you need 3,000 times more A beta 40 to get the same degree of inhibition. Okay. All right. Okay. So we showed this a variety of different ways. Red shows A beta 42, and you can see it completely shuts down degradation of a certain assay. It's an A beta degradation assay. 40 does hardly anything. 42 is usually around 1/10 to 1/5 of the concentration of A beta 40. So maybe this effect gets washed out because it's such a minority of the A beta species. No, if you add 1/10 the amount of A beta 42 versus 40, it actually substantially inhibits the degradation of all amyloid and other substrates. But here's the really important one that I want to focus on. Remember that cathepsin D degrades tau, which forms the tangles, the intraneuronal tangles. Well, what happens when you take tau and you put it with cathepsin D? Over time, it begins to degrade the protein. You can see this dark line getting lighter. You add a certain amount of A beta 40, and you get basically the same amount of degradation, but you add the identical amount of A beta 42, and it completely shuts down tau degradation. Okay, so we've recently extended these studies to show that this, I'm going to call it length specific inhibitory effect, right? If it ends at 42, it's strongly inhibiting. If it ends at 40, it's not. Well, the first thing we found was that is that if we take full length human A beta, is what we're doing all along, versus mouse A beta, which does not aggregate, we see the same effect. We see complete inhibition by 42 and partial inhibition with 40. But what about this thing called the P3 peptide? This is the AP amyloid precursor protein molecule. 90% of it is processed at the different site that cuts right in the middle of amyloid and prevents amyloid from being produced. So there's 10 times more of this little peptide that's like half of A beta. So we call it 17 to 42. And lo and behold, it has the exact same effect. So that really points to this red cleavage site as being the important one, because that's what's common to all of these peptides. But what we did is we generated a huge library of peptides that begin at different positions. So they begin at, if I look up here, like 17, 20, 21, 23, 26, 28, 30, 31, 32, something like that. And then we varied the ends. So 42 would be ending at this A, 41 would be ending at this I, 38 would be ending at this G, and there's another one which is 43. So we did 5 different lengths, and you can see that it doesn't matter where it starts. The big important thing is the number of amino acids on that little piece of the amyloid protein. So here's a summary of all these results. If 42 has a certain inhibitory power, well, 38 takes 700-fold more to have the same inhibitory power. And you get this effect where it's like longer peptides up to 42 become more inhibitory. And then when you go one more amino acid, that interrupts it. So we think this is a very specific, highly detailed molecular mechanism that we're dealing with here. And maybe we can, capitalize on it to develop therapies. So I have a collaboration with Dr. John Brunning and Jordan Peterson in Australia. We're trying to get the crystal structure of the cathepsin D molecule with, we tried to develop some inhibitors that would, so they go up the enzyme in a way that really works for this. So these are actually new 3D crystallographic data, and they're overlaid on an existing structure. So we got beautiful data. We just didn't get one with the inhibitor. We're actually going to be doing that this month. So hopefully something comes out of it. Okay. So, but here's the important thing. Couldn't A beta 42 exert its pathogenic effect? Why is 42 so important? Because it aggregates more? I'm not so sure, because there's evidence that the aggregates outside the cell are maybe even helpful, because you're getting this amyloid out of the way. It's getting just packed into these plaques where it can't do anything. But could it be that the pathogenic effect of A beta 42 in Alzheimer's disease acts via inhibition of cathepsin D? So we normally think, oh, okay, you got these mutations, or you got aging and oxidative damage and all this stuff, and that reduces the activity of a protease, which increases amyloid. Well, I'm talking about the opposite. I'm talking about, well, maybe this increase in A beta 42 causes a decrease in cathepsin D activity, which causes downstream consequences. And particularly, what about tau? Okay, so we decided to look at a bunch of other amyloid, Alzheimer's disease related endpoints. And so here's the wild type animals in the top row, normal mice, and then cathepsin D knockout in the bottom. And you could see, here's cathepsin D. You can see it's expressed in the wild type animals, the normal animals, and not expressed at all in the knockout. Well, here's A beta. You don't see any A beta in the normal animals. You see a ton in knockouts. We knew that. Ubiquitin's another protein that accumulates in these neurodegenerative diseases, and it's massively increased. GFAP is kind of a marker of some kind of inflammatory response. It's also upregulated. But then we stained for this silver stain called Gallius, and you never see any staining in basically any animal model of Alzheimer's disease. But these knockout animals at 3 weeks of age had this humongous increase in this Gallius silver staining. So when you zoom in and you look at what this looks like, this looks like that perinuclear staining that we saw where amyloid accumulates. And guess what? It looks almost identical to what happens in human Alzheimer's disease. These are the tangles. These are the tangles stained with the exact same stain in a 77-year-old Alzheimer's patient. Here is with the same stain in a 25-day-old mouse. Okay, so that led us to look, well, what's going on with tau in these animals? And we found this remarkable increases in the phospho-tau, which is the kind of tau that forms the tangles. It's the brown staining in the cathepsin D here. And guess what? It accumulates in the same sort of loci as you see in the silver staining. And we also did a control where we looked at another animal that dies prematurely, which is another lysosomal disease. So it causes problems with the lysosomes. Nothing in those animals, even though they were collected at the same few days before death. If we look at another very well-established stain for PHF1, you can see it's massively upregulated in the cathepsin D mice. Okay, so what about at the protein level? Oh, sorry. At the protein level, this is total tau. Now, if you quantify total tau, it's not that there's more tau, like there was more amyloid in these animals, but it's more like there's a qualitative change. And these higher species up here, these ones that are higher in this particular Western blot, those are hyperphosphorylated species of tau, which is exactly what happens in Alzheimer's disease. That could be verified by staining for a phospho-tau antibody like this. And you can just see this huge increase in phospho-tau in these animals, this wild type knockout. And there's another form of a caspase cleave form, but okay. But the definitive biochemical hallmark of true tauopathy, true tangles, is what we call sarcosylin-soluble phospho-tau. So that's just a detergent, and that's what we do to sort of detect neurofibrillary tangles. So if you look at wild type animals, we're looking at a soluble fraction where the proteins can float around in the water-based medium, or pelleted material, which when it aggregates, it tends to go to the bottom under centrifugation. And we looked at wild type animals, cathepsin D knockout animals. Then we looked at a true animal model of tau, where these animals get robust tangles. They're so sick at this age that they are paralyzed in their cage. This is a 9-month-old animal. And then we looked at that other lysosomal dysfunction model, crab A mice. And as you can see down here, this is the money graph. There's way more phospho-tau, sarcosylin-soluble phospho-tau, than there is in this deliberately manufactured high tau expressing animal model. By 3 weeks of age, this is what happens in these animals. So this really does seem to imply that cathepsin D is involved in tau clearance and tau dysfunction. So we use this interesting cell model where we could turn on tau and turn it off. So we turned it on for a few days, and then we would turn it off, and then we would collect protein on the day we turn it off, 1 day, 2 days, or 3 days later. And we did this in the presence of a cathepsin D inhibitor, pepstatin A, or a control. And as you can see from this chart, basically the half-life of tau went from less than a day to a full day. So that means it really is seeming to impact tau even in a system like this. Now, I want to emphasize something. I told you about these mice that I made in 2003, which showed absolutely zero amyloid accumulation. Well, people used to ask me, well, what does this do to learning and memory? And I'm like, well, why would I bother, right? There's no plaques in these animals. Like, obviously, it's going to completely eliminate all the amyloid-related problems, right? Well, I was contacted by Leonard Mukey at UCSF, and he got those animals, and he did the experiment. Well, guess what? These animals here, which have absolutely no plaques whatsoever, but they have neprilysin overexpressed, right? So it's knocking down the amyloid. They are as dumb, or are as impaired, shall we say, as the mice full of amyloid. So what is this telling us? So we have all these therapies which are targeting extracellular amyloid, and yet when you eliminate extracellular amyloid, it has no effect on learning and memory. There's other examples of this where, like, Todd Goldie expressed amyloid exclusively in the extracellular space outside the cell. Those animals had no impairments whatsoever. So plaques don't cause memory problems. Maybe extracellular amyloid causes no memory problems as well. So we are trying to address this by generating. This takes so many years of time, but we've developed a beautiful system where we can down-regulate cathepsin D in a titratable manner, right? So that means we can determine by adjusting the dosage of a drug how much we down-regulate it. And we can also do it reversibly. We can remove the drug and it goes back to normal. So now we finally will have, by about the end of this year, a super valuable animal model to finally really investigate what cathepsin D is doing in the absence of this premature lethality. So we'll be able to cross them to animal models of amyloid and animal models of tau and see what happens. So that's the future directions. I'm comparing the downregulation of cathepsin D, which increases intracellular amyloid, to the downregulation of neprilysin, which increases extracellular amyloid. And we're asking, what's the relative importance of that for Alzheimer's disease pathogenesis? So, to conclude, by several measures, cathepsin D appears to be the principal A-beta degrading protease. And I think this really uncovered something people weren't aware of. Yeah, there's secretases, but what about the amyloid that was generated in the endolysosomal system? About half of it probably goes to the lysosome. And so it looks like a large portion of newly synthesized A-beta goes to lysosomes, and we weren't even aware of this. It degrades A-beta 42 and 40 with markedly different kinetics. A-beta 42 inhibits cathepsin D extremely potently. I mean, I wish I had an inhibitor that was that strong. Deletion of cathepsin D results in pronounced tauopathy by just 3 weeks of age in the absence of any overexpression. So the hypothesis is that maybe increased A-beta 42 in Alzheimer's disease contributes to the tangle formation by inhibiting cathepsin D and its clearance of tau. So I have many people to thank, including CART, of course. And here is just another mention of the nonprofit that I started, August Dieter Foundation. It's got kind of a weird website. It's not .org, it's .foundation, AD.foundation. So if you're interested, you can check it out. Thank you very much.
Thank you, Dr. Leissring. We really appreciate that We have just about 3 to 5 minutes left. If someone has a question for him, we will be happy to entertain that.